No worries at all. So I'm Dolly Jennings, and thank you guys for inviting me to give a journal club presentation on my latest paper that has come out. So I am currently a doctoral student at Florida State University in the Biomedical Sciences Department and in the program in neuroscience. So yeah, I'm looking forward to discussing this paper today. And at any moment, if anyone has any questions, you can interrupt me or you can save it for the end. And I am aware that, some of you guys or most of you guys are clinicians. And so I'm on the preclinical side. So if there's anything that I don't explain thoroughly enough as I should, just let me know. Um, okay, so this paper is examining the effects of chronic alcohol consumption and chronic social isolation stress on the reinforcing properties of ketamine in male and female long Evans rats, and these are adult rats. Um, so first, I'm not going to get into the background of major depressive disorder, alcohol use disorder. You guys all are aware of what these disorders are and the use of ketamine specifically in depression. But the main objective of this study was to examine the repeated use of ketamine in animals that were also exposed to chronic alcohol consumption. So we know that there's high comorbidity with major depressive disorder and alcohol use disorder. And so we're curious to see, is ketamine safe in individuals that may have these comorbid disorders? And additionally, we wanted to look at sex differences and the effects of isolation and alcohol on the reinforcing properties of ketamine, because we've shown in our lab that in female rats, these rats are more susceptible to ketamine's antidepressant effects, as well as rewarding and hedonic effects at much lower doses than males. So we also wanted to look at biological sex as well here. So again, overall objective, determine how a history of social isolation, alcohol, and biological sex influence the reinforcing properties of ketamine. So I'll go into this timeline in much greater detail in the next few slides. So first, we used chronic social isolation. So depression, as we all know, is an isolating disorder, and extended periods of social isolation can increase vulnerability to developing depression. And so we chose to use chronic social isolation in our rodents, as this has, it's an ethologically robust stressor in rodents, in both males and females. And it does result in behavioral and physiological manifestations of anxiety-like and depressive-like symptoms that kind of model the human conditions. Although we are aware this is not a model of depression. We say depressive-like or anxiety-like. So this model has shown that when treated with something like ketamine, there's a reversal of this depressive-like state that is induced by social isolation. And so all this evidence supports the preclinical validity of using this model here. And so the way that this works is that, again, we're using adult male and female long Evans rats. So we have our socially isolated group, and these rats are single housed. And of course, they have, access to food and water, environmental enrichment, and normal bedding in their cages, but they are single housed, and there are dividers up that are preventing them from seeing the neighboring cage mates or neighboring rats, so to speak, on the racks. And our pair housed group, of course, they're in the cage with a buddy. This is same sex pairing, so males and males, females and females. And they're able to, kind of socially interact with the neighboring rats, look at them and whatnot. So importantly, these animals are in these respective housing conditions throughout the duration of the project. So this is, again, chronic social isolation. So next, I'll get into our alcohol drinking paradigm. So we use something called intermittent access to two-bottle choice drinking. This is an established alcohol drinking paradigm in rodents. And again, we wanted to see the combination of social isolation and alcohol on the reinforcing properties of ketamine. So repeated cycles of intermittent access allows for repeated consumption, followed by withdrawal and forced abstinence cycles. And this reliably produces consistent consumption of whatever concentration of alcohol you're using. It's typically 20% ethanol. without using things like water restriction and sucrose fading procedures. And this is across sex, species, strains, and over long periods of time. It also has shown that it induces depressive-like and anxiety-like states in rodents. And importantly, the animals do get the choice between water or alcohol. And so it kind of models the human-like conditions of choosing to drink. And so the way that this works is we did have a period of 12 weeks where the animals were undergoing intermittent access to two bottle choice drinking. The drinking sessions were 4 hours long on Mondays, Wednesdays, and Fridays. So of course we have our alcohol drinkers that receive a bottle of water and 20% alcohol. And then we have our water controls that just receive 2 bottles of water. And so the way that this works is that at this onset of the dark cycle, which is when the rodents are most active, we remove the animals out of their home cages and we place them into a designated drinking cage that's similar to their home cage. We just want that new environment associated with drinking. Importantly, we do have to separate the pair-housed animals into their own cage as well, so we can measure the individual intake and consumption and preference for alcohol. But because the sessions are limited to 4 hours on 3 days a week, we're not worried that the social isolation effects will be induced in the pair-housed rats during these drinking sessions. So remove the animals from their home cage, place them into their drinking cages, place the bottles on, and then they drink for 4 hours. At the end of the 4 hours, bottles are removed and we weigh them and we can measure both preference for alcohol over water as well as consumption of alcohol. Okay, so as I said, there are a total of 12 weeks of intermittent access drinking. So following the 11th week of drinking, the animals undergo jugular catheter surgeries where this subcutaneous backport is placed for intravenous drug delivery. So there's a tube placed into the right jugular vein, so the animals receive direct intravenous access when they make the correct response here. And so the way this works is that here's our operant box, and we have this magnetic tether, as you can see over here in the rats. It's connected to this backport that I mentioned. And there's like a swivel in the boxes where the animals are able to freely move around. They're not restricted. They're able to kind of explore within the box. And so following these 12 weeks of drinking, the very next day, so the last drinking session is on a Friday, and then on Saturday, the animals begin the ketamine self-administration portion of the experiment. And so similar to the alcohol drinking portion of the study, at the onset of the dark cycle, this is when the self-administration sessions begin. So the animals are brought into the room, they're placed into the respective operant box, and these sessions last 2 hours. And so the way that this works is that, again, the animals are placed into the box, connected to this tether. Here we have pumps that deliver ketamine through this tubing, through the animal's backport, and through the catheter, of course. And so at the start of a session, the house light is on, and if the animals make a correct response, which is going to the active site nose poke hole, a cue light comes on at the same time, and as well, 1 infusion of 0.5 mg per kg ketamine is delivered. So this is a fixed ratio Schedule 1 reinforcement where they have to make 1 response to the active site nose poke hole, and they receive 1 infusion of that 0.5 mg per kg dose of ketamine. Following that infusion, there's a 20-second timeout period where they can still respond to the active and inactive site nose poke holes, but they do not receive infusions of ketamine. So we're still recording these responses, but they have to wait the 20 seconds to receive another infusion. So at the end of that 20-second timeout period, the house light comes on again, signaling to the animal. They go to the active site nose poke, cue light comes on, they receive an infusion of ketamine. If they go to the inactive site nose poke hole, they don't receive any cues, and of course they don't receive the infusion of ketamine. And so as you'll see, they very rapidly learn what side gets them ketamine and what side does not. So following the 10 sessions of acquisition, they then undergo 10 sessions of extinction training. And so what happens here is that we remove ketamine itself, of course. We also remove the cues associated with ketamine. So the sound of the infusion pump, again, ketamine itself, the house light, and the cue light. So all cues that were previously associated with ketamine are now removed. And the goal of this is to see that if we remove the drug and we remove the cues associated with the drug, do we see a reduction in responding, which would be here, a reduction in going to the active site nose poke hole. So this can kind of be thought of as if an individual goes into treatment, they're no longer using the drug, they're no longer exposed to the context and the cues in which they were using, with the ultimate goal of reducing those drug-seeking behaviors, of course. So following the 10 sessions of extinction training, the animals then underwent 1 session of cue-induced reinstatement. And so what happens here is that they still no longer have access to ketamine, but we return all of the cues previously associated with ketamine. So the sound of the pump, the house light, the cue light. And the goal here is to see that in the presence of these ketamine-associated cues, do we see a return of responding, which is to the active site nose poke hole. So again, this could be thought of as maybe if an individual leaves treatment and then they're back into the environment that they once used in, they're surrounded by these cues that then may trigger those drug-seeking behaviors. So that's kind of what we're trying to model here. Immediately after this single session of cue-induced reinstatement, the animals are then terminated and we remove their brains, and I'll talk to you about what we do with the brains. Okay, so to begin, what we are looking at here is a dendrite, and these little protrusions coming off of them are dendritic spines. So dendritic spines are small protrusions on neuronal dendrites. They serve as primary postsynaptic sites for most excitatory synapses in the brain. These dendritic spines play a crucial role in receiving and processing vast amounts of information between neurons in the brain. And so what can happen with learning and drugs of abuse is that we see structural changes in these dendritic spines that of course then lead to functional changes. We also see changes related to these structural changes in neurotransmitter receptors. So changes in the number of these receptors and then also the function of these receptors, which ultimately leads to an overall change in function of neurons and entire circuits as well. And so what we wanted to do is we wanted to use something called the dialytic labeling of dendritic spines. So we wanted to look at the nucleus accumbens, which is an area involved in reward and implicated in chronic drug use and compulsive drug use as well. And so what I do is I make these little bullets here that contain this dye called dye I, which fluoresces red. And so I'm able to slice the brain, which is it's fixed tissue, slice the brain, and then whatever area I'm interested in this case, the nucleus accumbens, I then load these bullets into this gene gun and I'm able to shoot the region I'm interested in and looking at, and it will fluoresce red. And then I use high resolution confocal microscopy to then image these dendrites and dendritic spines. So we're looking at these different spine subtypes here, which can be a measure of the types of changes that are occurring following chronic alcohol isolation, differences in sex, and then ketamine use as well. So Philopodia is involved in they're less mature spine subtypes, but they're involved in initiating contact with presynaptic neurons and the induction of spinogenesis and synaptogenesis. So new spines, new synapses. What I want to mainly focus on is the long, thin spines and the mushroom spines. So long, thin spines are also considered immature spines. They have less developed postsynaptic densities. This means that they have less of these neurotransmitter receptors. So in our case, we might want to consider the glutamatergic receptors, NMDA receptors, which of course alcohol and ketamine are non-competitive NMDA receptor antagonists. So this might be something we focus on here. And so these long, thin spines have less of these receptors, but through learning and drug exposure in this case, they can then become these mushroom spines that do have fully developed postsynaptic densities, including these neurotransmitter receptors. And so importantly, measuring these changes in dendritic spines. So following drug use, this leaves the brain, of course, in this enhanced state of plasticity, which can manifest as changes in dendritic spines and receptors. And this can manifest as things like tolerance, withdrawal, and continued drug seeking. So that's kind of why we wanted to examine changes in spines. We wanted to see the interactions again with isolation, biological sex, and alcohol. So now I'll get into the results. So first we'll discuss the alcohol drinking. So again, 12 weeks of intermittent access to bottle choice drinking, 4-hour drinking sessions, 3 days a week. And so here when looking at, we have alcohol consumption measured in grams per kilogram at the top, and then at the bottom, we have preference for alcohol over water. We have our males on the left and our females on the right. And what we see is that overall, we see that females consume more alcohol and prefer alcohol more so than males. This is a pretty robust finding across different species and strain with different drinking protocols as well. So we weren't too surprised to see this, but nevertheless, females consume more than males. We did not see any effects of isolation on alcohol consumption. What we did find, though, is that in our males, in our isolated males, they did tend to prefer alcohol over water more so than our pair-housed males. And we have found in the past in our lab and in others that male rats tend to be more susceptible to the effects of social isolation compared to females. So this kind of supports those the previous findings as well. So next, we're going to get into the self-administration portion, so the acquisition training. Again, we're using an FR1 schedule of reinforcement. They respond once, they get 1 infusion of 0.5 mg per kg ketamine. We're going to focus on the infusions up here. As I mentioned, during those periods of timeout, we do record the active and inactive site nose pokes, but we really want to focus mainly on how much ketamine they're receiving. And so in our males, we see that in our isolated water controls and these lighter blue triangles, we see that they're receiving more ketamine than our pair-housed water controls, again, supporting the notion that males are more susceptible, at least in this model, to the effects of social isolation. We do see, however, though, that once there's a history of alcohol added, we no longer see that effective housing. So it's as if alcohol is causing some type of compensatory effect almost. So we see that in our alcohol drinkers, there's a strong trend for these animals to take more ketamine than the water controls. In our females, this is even more heightened. So we see that in our females that have a history of alcohol consumption, and again, these females were consuming the most out of any other group in terms of alcohol, they also receive the most ketamine out of any other group. So overall, a history of alcohol consumption in both sexes increases the reinforcing properties of ketamine, as seen here by how much ketamine they are receiving. And also in males, a history of isolation increases ketamine intake. I do want to comment on the inactive site findings down here. So overall, it's clear that they are able to discriminate between what side gets them the drug and what side doesn't. We see more responses to the active site compared to the inactive. But what we did find is that in our males that are isolated water controls over time, and in our females that are alcohol drinkers over time, we see that they do respond more to the inactive site. And as we know, ketamine has cognitive effects. And so it could be that the cumulative effects of these animals are receiving a decent amount of ketamine in these sessions over these 10 days, that there is some type of cognitive effect happening where, yes, they're still responding more at the active site, but they do have some of those incorrect responses at the inactive site as well. So here, this is looking at our extinction training. So again, what we do here is we remove all of the ketamine-associated cues as well as ketamine itself. And the goal here is to see if we have a reduction in active site responding. And as we see in both our males and females within all of the groups, we see that when we remove the drug itself and the cues compared to our acquisition day 10, we see a rapid reduction in responding. So this is showing that, yes, these cues are powerful enough to associate responding, and that when we remove them, we no longer see that responding. Now this is looking at our cue-induced reinstatement session. So what we find here is that in all groups and in males and females, when we look at that return of responding to those ketamine-associated cues, but we do not give ketamine, just the cues, we do see a return in active site nose poke responding. So we see a return of these ketamine-seeking behaviors. And we see this, that all of the groups, regardless of housing or drinking history, do reinstate to these cues. In females, we don't see any differences between groups, but in the males, interestingly, we do see that the animals that were exposed to a history of isolation and alcohol actually have a higher magnitude of reinstatement, so increased ketamine-seeking behaviors in this group compared to that group that was pair-housed with alcohol only. So next, we're looking at our dendritic spine findings. I want to mainly focus on our long, thin spines and our mushroom spines, as I mentioned before. And so in our males, we see that kind of similar to what we see with ketamine acquisition, is that, and alcohol, is that we have an increase in our long thin spines and our isolated water controls. And then interestingly, when we add a history of alcohol as well in these isolated animals, this actually causes a reduction in these thin spines, or is associated with a reduction. We did not see any changes with our mushroom spines in our males. In our females, though, we see that there's an increase in our long, thin spines in our alcohol drinkers. And then in our mushroom spines, we see that this was affected by housing and drinking. So we see that a history of alcohol in our pair-housed animals reduces our mushroom spines, so the more mature spines with more receptors, which kind of makes sense given our increase in thin spines that are less mature. And when a history of isolation is added, though, we see that there's an increase in these more mature spines that may have more receptors. Okay, so some of the conclusions from this work. So alcohol may increase the reinforcing effects of ketamine in both our adult male and female Long Evans rats. As we saw that our alcohol isolated animals, or our males specifically, did have an increased rate of reinstatement to ketamine cues following a period of without ketamine or the cues. We saw that this increased reinstatement, and so this may indicate that in males that have comorbid depression and alcohol use, following treatment or even during treatment, they may be at an increased risk of ketamine-seeking behaviors. Overall, this may suggest that ketamine treatment doses may need to be adjusted to account for what could be happening is that potential cross-tolerance of alcohol and ketamine. So we know that chronic alcohol has effects at the NMDA receptors, and we know that chronic ketamine also does. And so what may be happening is that it's not necessarily an increase in the reinforcing properties of ketamine following alcohol, but that there's some receptor change that occurs with chronic alcohol that then may influence how the individual responds to ketamine. So again, this is kind of what I've said. So it could be that alcohol-induced NMDA receptor desensitization could influence responding for ketamine in these individuals with a history of alcohol use. This could influence the therapeutic effects. So increased doses of ketamine may be needed to experience these effects, or it could also reduce the aversive effects of ketamine. So I just want to focus on that here because, we can't fully say that alcohol is 100% increase in the reinforcing properties. It could be more at play here than just simply that. And then again, related to the changes in NMDA receptors that may be happening is we do see changes in these dendritic spines are still sustained well into abstinence. So we did see that 3 weeks into abstinence from chronic alcohol and 10 days into abstinence from ketamine, we see these changes in spines. So this suggests that there's an enhanced state of plasticity that remains in the nucleus accumbens following the cessation of alcohol. Little caveat here is that we did not have saline controls, and so we can't really compare, is this, we can really only say compare the alcohol and water controls, and of course paired and isolated animals, but we didn't have that saline control to compare with the ketamine animals. And so in the future, we would like to see, we would like to include saline controls to see if there are any differences, which we imagine there will be. And then also the timing of changes. So it could be that maybe it would be useful for us to terminate the animals at different time periods during chronic drinking and ketamine treatment to then see, well, what changes are happening at this time point, maybe an earlier time point into withdrawal or abstinence, and then how does this correlate to behavior. And then we would also like to look at, so we have seen in looking at the antidepressant effects and hedonic and rewarding effects of ketamine. As I mentioned, females respond at lower doses than males. And we have seen evidence that this may be due to cycling ovarian hormones. So females in proestrus respond similarly as males, but when females are in diestrus, which are lower levels of hormones, we see that there's just differences in how hard they're willing to work for ketamine and the rewarding effects of ketamine. So there's also evidence with chronic alcohol that estrus cycle may be disrupted or just the role of ovarian hormones in and of itself could influence responding for alcohol and ketamine. And so we would like to also look at estrous cycle and responding in this model in particular. And that wraps it up. If anyone has any questions regarding this work or anything else, my email is up here. And I'll be defending my dissertation late May. So I'm also on a job hunt currently. So if anyone is interested in checking out my LinkedIn profile, or again, any questions, I'd love to answer those. But thank you again for inviting me to share my work. I was very excited for this. And yeah, I can answer any questions.
That was stunning, Dolly. Thank you. Really, really nicely presented, really clear. Just fantastic.
Thank you.
A lot of it. A lot of it. And so a little bit hard for a clinician to sort of try and get my head around. I mean, I'm particularly interested in this because we're currently doing a study looking at the possibility of ketamine as a treatment for people with to maintain abstinence in people with severe alcohol use disorder. So you probably know of Celia Morgan's work at Exeter, where he randomized trial. So that phase 2 trial was completed and suggested that ketamine reduced alcohol consumption at 6 months, particularly if it was paired with psychotherapy. And so we're doing a phase 3 study at the moment. We're recruiting for it to try and replicate that.
I'm very excited to see that work because I have been following that, so I'm very excited.
Yeah. What would you predict?
So I guess my biggest concern, and again, I'm looking in the animal models, but what I would also want to do, and I've kind of put some of this in my dissertation for future directions. Obviously, that clinical work is suggesting that ketamine is effective in increasing days abstinent. I guess the concern would be two things that come to mind. If we're not seeing, because I know some people have seen reductions in the heavy, harmful drinking days, my concern would be, well, if we're still seeing the presence of that, could that, just result in full-blown relapse? And then at what time point is a ketamine maybe booster session needed to prevent that, or if it does happen, to then increase abstinence following that relapse. Another thing would be maybe because, ketamine feels like alcohol, I guess the concern would be, would that in and of itself reinstate alcohol drinking? Is that's what I've wanted to model in my animals is, following a longer period of abstinence, if we give ketamine and then alcohol, what happens with that? Does ketamine almost reinstate drinking? Or alternatively, and the positive side of it, of course, would be because it feels like alcohol, they don't have the need to drink, which I hope is obviously the side to that story. But yeah.
I really understood what the second of those two things that you were saying. So your prediction would be that, or your fear is that, say they have these ketamine through 3 ketamine infusions, and then 6 months later you'd be worried that their alcohol use was actually greater. Yeah, okay. And then the first bit, I was less clear about what you were saying with the booster session and when the booster session was needed. Can you just go over that again?
Yeah, so I guess actually the second paper I'm working on now, hopefully that will be submitted in the next few months. We looked at different types of drinking models and then when to give ketamine during different time points into withdrawal or abstinence. And I guess, part of what I found is that in females, the effect lasts a bit longer than in males. And so what that work is kind of inspiring me to, I'm curious to see is how long the effects last. And of course, Celia Morgan's work showed that they are abstinent at the 6-month follow-up. But yeah, I guess how long, of course it'll probably be affected by individual differences, I'm sure too. Maybe when someone would need like a booster session if they start experiencing craving, maybe 3 weeks after an initial session, or that's kind of what I meant there. If does that make sense?
So you would have to time a ketamine infusion to match the time of craving for alcohol, in order
Mm-hmm.
that ketamine is essentially substituting for the alcohol and is just
Mm-hmm.
a safer
Mm-hmm.
drug, perhaps. Is that what you're saying?
Almost, yeah, almost like, I guess methadone and opioid use. So kind of like that replacement therapy.
Okay, right. No, I'm with you. Yeah, yeah.
Sorry.
No. That's great. No, that's really helpful. So obviously when we give people ketamine, they have a sort of dissociated experience. They're a bit out of it. Sometimes I'll say to them, some people describe it a bit like they're feeling drunk. Obviously, there are substantial differences as well. So at the sort of synapse level and the spine level, the effect of the ketamine and of the alcohol, they're sort of broadly similar. Is that what you're saying?
Yes. Yes. Especially with the repeated chronic use. And,
Yeah.
there's some debate, of course. That's kind of why I wanted looking into more of the timing effects, right? Because we don't know how long these these effects are lasting in spines. And again, with my work, we couldn't parse apart whether it was alcohol and ketamine, the combined effects, because we didn't have our saline group. So, also looking at that as well.
Great.
Sorry to interrupt. I think we have a couple of questions in the
Yeah.
chat.
Mm-hmm.
Sorry, just in the interest of time. And Rajan and Kwang, feel free to just like join us and ask the question directly if you feel comfortable, or we can read them out maybe.
Sure. Can you hear me?
Yeah.
Can. Hi.
Yeah, nice presentation. Thank you.
Thank you.
Yeah, I mean, there's a very interesting discussion. So, I was thinking about in those alcohol-dependent animals, but by the time when you tested, ketamine Q reinstatement, it was a while ago when animals, consumed alcohol. So if you
Mm-hmm.
let them drink alcohol before you do ketamine Q reinstatement, what do you expect?
Very, very good question. So this is something that we have also talked about doing. I would expect an increased rate of reinstatement from what we saw, just given the overlap in the effects. But also someone asked, is the concentration or type of alcohol has any impact? So that's kind of what we're doing now, is seeing the different lengths of exposure to alcohol. So overall, how much alcohol they're receiving. We tried to model the human conditions, where we have chronic drinking followed by periods of abstinence followed by relapse, to see the effects of treatment response. But again, back to your question, I would expect that if we gave alcohol before cue-induced reinstatement, given the overlapping effects, again, the dose of alcohol that we give and whether it's maybe experimenter administered or if they're choosing to drink, that would probably also play a role. And it's a little hard with alcohol self-administration and ketamine, at least intravenous self-administration, it would be a little hard because you typically don't give intravenous alcohol. It would be like an IP injection, intraperitoneal injection, sorry. So that might have some effects on how reinforcing it is as well. They're receiving, which they would receive the effects of alcohol relatively quickly with the IP injection as well. But different routes of administration might affect it. And then another thing that, we kind of had to note is that these animals are not dependent. So there are ways to induce dependence-like states in rodents. It's called chronic intermittent ethanol vapor exposure. And so they're getting blasted with a lot of alcohol for 16 hours straight. And what we see is that compared to before this vapor exposure, they're choosing to drink higher amounts than before they underwent that exposure. And so this is considered in the preclinical alcohol world a model of dependence. So we would also like to implement that model in a study like this to see, well, you know, because we can only really say in animals exposed to alcohol, right? We can't really relate it to dependence or alcohol use disorder. So using that model would really provide even more insight into the dependent side of things.
Yeah, very interesting. Thank you. Just one more question. As you know, the more animals take ketamine, they get dissociated, right? So they show like dissociative symptoms, especially with the IV ketamine self-administration. So, like, what do you think about that dissociative side effect? Because I guess the more they take ketamine, they will show more dissociation.
For sure. And we see that with our the behavioral, the locomotive effects as well. So over time, we see that there's that sensitization effect. And even just taking my animals out of the operant boxes, they were mainly the females. They did take the most ketamine. They were visibly impaired. So, you know, I think maybe that could be what sets apart alcohol and ketamine, where you wouldn't have, like how my concern was, that ketamine could almost reinstate alcohol. But then again, my concern would be if we do have those sensitization effects with chronic or repeated ketamine, you know, what is that doing to the brain? What is that doing to the receptors or the dendritic spines to, you know, maybe increase ketamine seeking in the individuals receiving treatment. What we are doing currently, I'm not personally working on it, but in the Cabage lab, we have someone working on more of the cognitive effects with repeated ketamine. So it'll be interesting to see. She's using things like set shifting. So it'll be interesting to see at least, you know, some of the cognitive related effects, kind of getting at your dissociative effects.
Interesting. Thank you.
Thank you.
We've got a question, is concentration or type of alcohol, does that have any impact? What would you expect on that?
So do you mean like in the human standpoint, like if it was like beer versus liquor or something like that, or do you mean in the rodent model where it's the dose that they're getting, or the like, you know, we use 20%, maybe we use 10% instead? That's probably more what you're asking for me.
Go for that one, yeah.
So this is kind of related again to something I've been looking at with the discriminative effects with alcohol and NMDA receptor antagonists. So the dose absolutely matters. So the higher the alcohol dose, the more it is similar to the NMDA receptor antagonist. So of course, ketamine included. So yes, dose absolutely matters. And you know, how long they're exposed to the alcohol drinking paradigm, of course, would also matter. So we do try to use more chronic paradigms. But in terms of just a dose association or discriminative properties, the higher the alcohol dose, the more likely or the more similar it is to the ketamine.
I know we are approaching the end of our time together, but I was wondering if I can just jump in very quickly with a question. So I don't know, I know that Rupert mentioned the work with Celia Morgan, but there's been also another work that has been conducted by DAS, where it was looking at, I don't know if you're familiar with it, but basically maladaptive drinking behavior and the use of ketamine associated with the memory reactivation of it. And your work made me think of that as well. And I know you focus primarily on the reward system, but I was wondering if you also look at other brain area, something like maybe more involved with maladaptive rewarding memory or anything along those lines, or if you have any thought about that.
So the only other region that I've looked at, and this is more in just looking at the therapeutic effects of ketamine in our different models of alcohol, is the prefrontal cortex. We were just looking at, I was looking at CFOS, so just neuronal markers of brain activity. And I looked at the amygdala as well, the bed nucleus of the striatum terminalis, and also the nucleus accumbens. Interestingly, and I think it's a methodology issue. We only found differences in the prefrontal cortex. And of course, we know that ketamine, there's more known in the depression world with ketamine and the prefrontal cortex. So another thing that I wanted to look at is because, at least to my knowledge, there's still a lot we don't know about, again, ketamine's mechanism of action in the alcohol exposed brain. So I wanted to look at somatostatin expression. As you know, there's a little bit more known out there on the depression side. And what I found, so with chronic alcohol, we see in the rodent model that there's a decrease in somatostatin expression, meaning that there's overall an increase in glutamatergic activities. That's why we see things like hyperexcitability and withdrawal, negative affective like states. So what I'm proposing ketamine may be doing is, because in my animals that responded to ketamine, so we saw a reduction in drinking, those same animals had an increase in somatostatin expression, suggesting perhaps that there's an increase in overall GABAergic tone, so creating that anxiolytic or antidepressant-like effect, and kind of, you know, reducing that hyperglutamatergic state. Again, complete speculation. We didn't do any functional work to follow that up on. I'm hoping once I graduate, someone else does that in the lab. But I don't know, that's kind of like where I've been putting my focus now, is looking specifically at the prefrontal cortex and the somatostatin expression. Now, in terms of the memory side, I am familiar with the paper that you're referring to, but in the animal side, I'm not familiar with, I don't believe anyone has really looked into that. But I mean, obviously very interesting. I know there's some preclinical work with ketamine and PTSD that might be more related to like what you're talking about with the kind of the rewiring of the memory.
Fantastic, thank you.
Anyone got any other questions? I'd just like to say how pleased I am that somebody's doing some animal work on the longer term use of ketamine and this sort of thing. I think it's really important, because for us clinicians, you know, the acute phase is dead easy. It's really how we start managing the long term and the tolerance that's induced by ketamine, and the reduction in the duration of the antidepressant spell. So anything that, you know, developing a model where we can model that the brevity of the ketamine antidepressant effect, and then using that as a basis for exploring ways of prolonging that. That's the holy grail. So I'm you know, I'm glad that you're sort of getting to grips with that long term use. So thank you for that. And
well, thank you guys. Sorry, go ahead.
No, go.
Sometimes I like, because I have a clinical background as well, and then I just switched to basic science for graduate school. And sometimes I often, you know, I can get bogged down in looking at this one little part of the brain and this one little thing in the brain and the one effect of ketamine or alcohol or whatever. And it's nice to speak with you guys and to hear that this work, you know, I always say like this suggests or this may imply, but it's nice to speak with you guys and see that, you know, this work does matter.
So just, I don't know if it helps, and it's sort of tangential really, but probably of all the papers that have influenced how I've thought about how ketamine works, the work of Hailan Hu has been the most significant. So it's animal data, you know, and I'm particularly obviously interested in rat data because they're just more social.
Yeah.
So I think, you know, all power to you.
Well, thank you.
Okay. Great. We've had some people enter late. I'm afraid that's probably an indicator of our problem that we've had with the clocks and so forth. But this fantastic talk will be available on our website within the next few days, I hope. So if you've arrived late, I'm very sorry about that, but you will be able to see this talk just in the next few days. So thank you so much, Dolly, and very best of luck to you in your career.
Thank you so much.
It's been great. Thank you so much. Okay.
Bye-bye.
Bye-bye.
Bye.